Hello everyone. So today's topic is about the cultivation of viruses. As we all know that viruses are extremely small infectious agent that invade cells of all types, and they are obligate intracellular parasites, which mean that they depend on host for their survival. They cannot be grown in non-living culture media or on agar plates alone. They must require cells to support their replication. Viruses have been the culprit in many human diseases, including smallpox, flu, AIDS, and the ever present common cold, as well as in plant bacteria and archaea also. And what are the techniques in cultivating and identifying animal viruses? Viruses require living cells as their medium. Okay, we have two techniques in vitro, and in vivo. In vivo, in vivo laboratory breed animals and embryonic bird tissues are used, while in vitro cells or tissue culture methods or techniques are being used. Okay, why we why we cultivate virus? What are the major purpose? The major or primary purpose of cultivating virus is to isolate and identify virus in clinical samples, to do research on their structure, replication methods, genetics, and effects on host cell, and to prepare virus for vaccine production. This is a very important point so that we cultivate virus. There are many viral vaccines. So you can see, okay, we can see later in this video. Okay, there are various types of vaccine involved in viral production, which is heat kill vaccine, mRNA vaccine, very famous in corona virus. Okay, let's move on. Okay, we already discussed the techniques and primary purpose of virus cultivation, and what are the methods of cultivating virus. Okay, here generally three methods are employed for the virus cultivation. Number one is inoculation of virus into animals. Here you can see inoculation of virus into embryonated egg or fertile eggs. Tissue culture. In this lecture, we will discuss tissue culture, what are their techniques and the types of tissue culture. There are three types of tissue culture and what are the effects, and later on cultivation of viruses or organ. For the growth of viruses, there are three types of tissue culture. First is your organ culture, that are mainly done for highly specialized parasites of certain organs. Example is your tracheal ring culture is done for the isolation of coronavirus. Explant culture, fragments of virus. Tissues can be grown as explant in plasma clot. This method is rarely used. And third is your cell culture. It is the mostly used method for the cultivation of virus. Let's see detail about cell culture. Cell culture is mostly used for identification and cultivation of viruses. Cell culture is a process by which cells are grown under controlled conditions. Cells are grown in vitro on glass or a treated plastic surface in a suitable growth medium. At first, growth medium usually balanced salt solution containing 13 amino acids, sugar, protein, salt, carb serum, buffer, antibiotics, and phenolate are taken and the host tissue or cell is inoculated. On incubation, the cell divide and is spread out on the glass surface to form a confluent monolayer. Now, what are the types of cell culture? Based on the region and the chromosomal property, the tissue culture are classified into three types. Number one is your primary cell culture. These are normal cells freshly taken from animal or human body. They are able to grow only for a limited time and cannot be maintained in serial culture. They are used for the primary isolation of viruses and production of vaccine. Monkey kidney cell culture, human embryonic kidney, chicken embryo cell culture are the major examples. Second is your diploid cell culture, which is a semi-continuous cell lines. They are diploid and contain the same number of chromosomes as their parent cell. They can be subcultured up to 50 times by serial transfer following senescence and the cell strain is lost. They are used for the isolation of some fastidious viruses and production of viral vaccines. Now there is a term of fastidious. We will later discuss it in bacteriology. The fastidious viruses or fastidious bacteria are fastidious organisms are those organisms that are not grown on their basic requirements. They had a very special nutritive requirement. If we provided these requirements, only those bacteria and viruses will grow on that media. So, diploid cell culture is used for the isolation of some fastidious viruses and the production of viral vaccines. Human embryonic lung strain, rhesus embryo cell strain are their examples. Third is your continuous cell lines. They are derived from cancer cells. They can be serially cultured, so named as continuous cell lines because they cultured serially. They can be maintained either by serial subculture or by storing in deep freezer at 70 degrees centigrade. Due to derivation from cancer cells, they are not useful for vaccine production. Examples are HeLa, human carcinoma of cervix cell lines, HEAP2, human epithelioma of larynx cell lines, BHK21, baby hamster kidney cell line. What are the advantages of cell culture? Relatively easy, broad spectrum, cheaper, and sensitive. Okay, there are some disadvantages of cell culture. The process requires trained technicians with experience in working on a full-time basis. State health laboratories and hospital laboratories do not isolate and identify viruses in clinical work. Tissue or serum for analysis is sent to central laboratories to identify bacteria. Okay, so this is today's lecture about cultivation. Hope you liked